there's a few reasons why I gave it to you. One is it's going to talk about primary and secondary endosymbiosis in the evolution of these protists. And so that's going to be a topic. There's a dynamite diagram on page 2 that cartoons primary and secondary endosymbiosis, which we will discuss. The other reason I wanted to give this to you is that it shows, I think on the last page, this is what's called a phylogenetic tree that traces the evolution of eukaryotes. And just to give you an idea of why this is also maddening, if you take a look at where the members of the old kingdom protists are located, they're sprinkled throughout all these branches of the eukaryotic organism. So we have members of Protista down with kingdom Animalia. We have members of kingdom Protista down with kingdom fungi. We have members of kingdom Protista down with the land plants. So what this tells us is that these protists, they evolve from many different lineages. What happened historically is that biologists didn't know where to put them. Most of them are unicellular eukaryotes, and the biologists were like, well, gosh, this isn't really an animal, it's not really a plant, it's not really a fungus, so we're going to dump all these unicellular eukaryotes into this kingdom Protista. So it's kind of a dumping ground, right? So, just so you all understand why classification is such a headache, and biologists are still arguing about it, and every year there'll be a classification change. So for that reason, we're not going to focus a lot on which phylum or which subgroup these protists belong to. Instead, we're going to do what is suggested by Campbell, and this is on page 2. I'm just going to read this to you guys. I was just like, yes! So they state down here, as you read this chapter, as we do this unit, it may be helpful to focus less on the specific names of groups or phyla of organisms and more on why the organisms are important. So that is going to be our focus, not to memorize which phylum or subgroup they belong to, but why are they important out in nature. So what we're going to do in classification today, we're going to use like a really old-fashioned way to classify them. We're going to use a classification scheme that's based on whether they can move or not, whether they're motile. And if they are motile, what structures do they use to move? So we are just so going to simplify this, and I think it's appropriate for us. Now, before we launch into a discussion of the 4 groups of protists, and just so you all know, the 4 groups of protists that we'll be discussing are the flagellates, the ciliates, and the amoeba. These are 3 groups we're going to discuss today. The 4th group, which are called the apicomplexa, this is the only group where the protists are not motile. We're going to save discussion of apicomplexa till Tuesday, just to break it up a little bit. Okay. What I want to do before we launch into these specific groups is to describe to you primary and secondary endosymbiosis so we can understand why the heck is this group so diverse. Okay? And to help with that, if you take a look at the Campbell handout on page 2, there's a dynamite cartoon. There's a dynamite cartoon of primary and secondary endosymbiosis. And I just want to do my own little cartoon up here just so we can understand. So again, we're trying to answer the question, why is there such incredible diversity amongst this group called the protists? How did it evolve? And hopefully we'll understand why biologists are still arguing over classification. All right, so let's see what I've got here. Oh, all right, so okay, so primary endosymbiosis explains the evolution of eukaryotic mitochondria and chloroplasts. Okay, so I'm just going to do this very quickly. Okay, so way back sometime, maybe like 2.6 billion years ago, there was a primitive aerobic respiring bacterium. This bacterium had an electron transport chain. It had the enzymes for glycolysis, Krebs cycle, electron transfer chain, ATP synthase. So these little guys, they were cranking out ATP like crazy. Okay? Now, at some point in history, and maybe more than one time in history, there was a primitive nucleated cell. So we could say it is a primitive eukaryotic cell, but these guys still had not evolved aerobic respiration. They carried out fermentation. And the story of primary endosymbiosis is that this primitive cell was a hunter, kind of like an amoeba. It phagocytized the aerobic bacterium. So put plus here. But instead of the respiring bacterium being destroyed, the respiring bacteria actually lived. It survived inside the primitive nucleated cell. Not only did it survive, it could multiply. Okay, so it would multiply. When the host cell, the bigger cell, divided, each of the two cells would acquire some of the little endosymbiotic bacteria. Now these little bacteria, remember, they're aerobic respiring. They're pumping out lots and lots of ATP. So the little respiring bacteria were supplying the host cell with ATP. And the host cell, because it was a hunter, it was supplying food molecules to little host bacteria. Okay. Now what did these little endosymbiotic bacteria evolve into after many, 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 many generations? The aerobic respiring bacteria evolved into what? Mitochondria. Right, exactly. So lots of time passes, right? And what we think is eventually those little endosymbiotic bacteria evolved into what? Mitochondria. Dynamite. Okay, so that is one example of primary endosymbiosis, the evolution of eukaryotic mitochondria. And it's thought maybe a little bit later, one of these primitive nucleated cells that already had mitochondria, it ate a primitive cyanobacterium. Okay, so again, many, many years ago, and let me see how I do this. Okay, so let's say this is our primitive cyanobacterium. So one of the nucleated cells that already had a little respiring endosymbiotic partner, or perhaps that had already evolved into a mitochondria, one of these guys ate a little primitive cyanobacterium. And what did that cyanobacterium eventually evolve into? Chloroplasts, right? Okay. So this eventually becomes a chloroplast. And I know I'm just making this really abbreviated. Okay. Primary endosymbiosis then explains the evolution of eukaryotic mitochondria and chloroplasts. Okay. Now, one of these early cells, and I'm going to wipe out this. Okay, one of these early cells that had acquired not only a mitochondrion, but it also had acquired a chloroplast. Okay, so we'll say chloroplast and mitochondrion. We're going to call this guy a primitive algal cell. Okay, so in secondary endosymbiosis, we have another primitive nucleated cell that eats an algal cell, okay, brings the algal cell inside. I know, isn't this crazy? So here we've got this guy, he's got his nucleus, he's got a mitochondrion, he's got a chloroplast. Okay, so here's the algal cell becomes an endosymbiont. Algal cell endosymbiont. This is an example of secondary endosymbiosis, where one of these eukaryotes that already had a mitochondrion chloroplast, again, was eaten by another cell, and it became an endosymbiont. Okay. Now, I don't want you to stress too much, you guys, about the secondary endosymbiosis. The whole reason for cartooning the secondary endosymbiosis is to help us understand why the protists are so incredibly diverse, why biologists are arguing and throwing mud at one another when it comes to classification. You can argue that some of the